Hello, everybody. Welcome back to a new D-Lab of Capital Cell. As you know, these webinars that we made in the private campaign of companies to introduce you the new opportunities that will be appearing Capital Cell website in the coming weeks. And today with us we have Mermaid Bio. We have Ranja and Chris. Ranja, CEO of Mermaid Bio, and Chris, the Chief Business Officer. From Mermaid Bio, which is a company developing a platform for antibodies against intracellular targets. It sounds really strange, and I think that Ranja and Chris will explain better than me what they are doing and what they can be the next unicorn of startups. And also with us, we have Klaus. Thank you to have it here. Klaus is an investor of Mermaid Bio, and he will explain also his career as an investor and CEO of companies, and why he decided to invest in Mermaid Bio. Nothing more to say from my part. As you know, the people that are registered here, we will do an explanation of Mermaid Bio, like 20-25 minutes, and then Klaus will have 5-10 minutes explaining why he decided to invest. And then we have a short time for Q&A. So if someone wants to ask questions, there's the tool of Q&A to write it down, but also there's a tool called raise hand, levantar la mano, which just means that if you raise your hand, we will give you the chance to ask the question in voice. With all that, Chris, Ranja, your time, please, and thank you.
Hi, everyone. So my name is Ranja. I'm co-founder and CEO of Mermaid Bio, originally from Germany, but as you may hear from my accent, I've been all around the world. And that's because early on, I actually focused on cell death and diseases associated with it, from inflammatory diseases and especially cancer. And that led me to Australia to do my PhD and postdoc. Shortly after that, I went into biotech because I wanted to be closer to the bedside of patients, where I worked in a company that focused on nanovesicles and drug delivery, be it DNA, RNA, small molecules, and so forth. And after a few years in that, I decided to do my own little stint, and this is why we're here today. And for that, I moved back to beautiful Munich in Germany. And I'm glad that I could convince Chris to join the mission.
Hi, everybody. My name is Chris Baldwin. I'm Chief Business Officer at Mermaid Bio, the other co-founder. I started my career in healthcare in New York at McKinsey way too long ago. And I've worked across the US, Australia, with healthcare companies, blood companies, plasma companies, medical device companies, biotechnology companies. And I met Ranja a number of years ago. And when she came up with the idea for Mermaid Bio and explained it to me in simple terms, which I needed at the time, I realized this was a once-in-a-lifetime opportunity. And so, of course, I was willing to pack my bags, move from Melbourne, Australia, with Ranja here to Munich, start the company 2 years ago. And we're really excited to share with you what that is. So I'll get the PowerPoint started, and I think Ranja will take you through it.
Right. So Mermaid Bio really is pioneering antibody-based precision medicine. And I'll lead you through what that really is. So antibodies have absolutely revolutionized the field of medicine because of their fantastic and perfect specificity, their fast discovery timelines, and also their limited toxicities. But actually, antibodies can only reach 10% of their potential targets, leaving many patients to not benefit from these therapies. And we, with our intrabodies, want to change that by taking antibody therapeutics to the inside of the cell so that we can reach the other 90% of the potential targets. And this is how it works. Really, what we do is we combine two cutting-edge technologies. On the one hand, we take lipid nanoparticles, which are famously known from the COVID vaccines, which are really just fat bubbles that have drug load that can be delivered. And on the other hand, we take new antibody modalities, so-called nanobodies or single-domain antibodies, and we encode them as mRNA and then encapsulate them into these lipid nanoparticles so that they can be safely transported in the patient's body to the organ and to the cell of interest. So when this LNP with the mRNA for the intrabody gets delivered into the cell of interest, the mRNA gets then translated into our intrabody protein. And then this intrabody can then bind to the intracellular target of interest and really take action. So what we have here is this dual specificity on the one hand from antibodies, together with targeted drug delivery, which gives us an amazing specificity and really creates a precision medicine approach that minimizes the off-target effects on the rest of the body of a patient. So our technology really works by a large platform, and this is how we make it happen. We identify high-value targets that we want to go after. And after we've identified targets, we have a rational antigen selection and design process, which will then identify the antigens that we use for immunizations in alpacas. Then once we put these in alpacas, what this gains us is all these different binders highlighted here in blue. All these different binders come from alpacas that we could potentially use as intrabodies, but we sift through them with various different technologies and assay approaches, as you can see top right, leaving us with only the really best candidates. These best candidates can then be further modified with our in-house technologies such as degraders, linkers, and cell killers to make them even more efficacious. So once we then have a final candidate, this gets encoded as mRNA and wrapped into LNPs so that it can then be delivered to the patient. So really what we're doing here with our intrabodies is having a precision medicine approach that is on the one hand incredibly tough on the targets, but at the same time really gentle on the rest of the body of the patient, leaving them unharmed. So with ample opportunity out there, where do you start? Since the intracellular proteome is pretty vast, we settled on targeting one of the big undruggables called beta-catenin. Beta-catenin plays a role in a lot of different cancers, but we decided to go after one of the most aggressive forms, which is hepatocellular carcinoma. We call this intrabody ABC, which stands for anti-beta-catenin. Hepatocellular carcinoma really is one of the most aggressive forms of liver cancer, and it doesn't have a lot of good treatment options on the market today. What it does have is really dire and terrible survival rates. What's more so, if you have a beta-catenin mutation in hepatocellular carcinoma, like 40% of the patients do, then you can actually not benefit from the first-in-line treatment, which is depicted here in the blue line, so-called immune checkpoint inhibitors. This is because beta-catenin drives resistance to immune checkpoint inhibitors, which leaves you with secondary treatments, small molecules such as sorafenib, which, as you can see, has really poor disease progressions. And in fact, a lot of patients choose to forego sorafenib because of its toxic off-target effects. So what this means is beta-catenin HCC really doesn't have many good drugs on the market today. And as you can see here in the bottom in these blue bubbles, if we only focus on the US market, our annual revenue is roughly below $4 billion US. So on a high level, how we build these intrabodies, over the last 2.5 years, we've really set up a stringent screening technique, and we've got a really vast in vitro data package now. When we have all these binders, we go through various different assays here, a Wnt signaling assay, to find the best inhibitors and the best binders for the job. We also look at their mode of actions and their functionalities by microscopy, Western blotting, and also by colony formation assays in the bottom. With microscopy, we can understand really how these intrabodies work and how they inhibit the intracellular proteins of interest. For example, in this case, our intrabody prevents beta-catenin, depicted in green, from entering the nucleus, where it would otherwise drive a lot of genes and lead to overproliferation and really tumor formation. We can also look at cancer cell lines like these two, HCT8 and H358 at the bottom, and what happens when we express our intrabody. And we can look at the survival of these cancer cell lines, like these purple colonies, which really depict tiny little cancers in a dish. After stringent in vitro screening, what we do next is look at in vivo models. And I'm sorry if you were about to have dinner. I hope you already had your dinner. But we work with mouse models since the HCC mouse models are incredibly close to the HCC human genome. So in our HCC model in mice, what was very clear when we injected our intrabody, what you can see here is that these mice develop HCC very rapidly and very aggressively. So when we don't treat with our HCC on the right, The livers get incredibly large. These mice form big nodules, and actually the ethical endpoint was reached because these mice were so sick that they couldn't move around anymore and they weren't feeding anymore. Whereas with our ABC treatment, we could significantly reduce the tumor development and the tumor burden. And in fact, the liver was intact and healthy and no other organ was affected at all. And the mice were not different to healthy mice. Apart from this in vivo, of course, which was a huge de-risking, the entire anti-beta-catenin project is heavily de-risked on a lot of different levels for us. Number one, beta-catenin has been around for about 3 decades. It's a well-established cancer target that's been trialed and tested time and time again. But a lot of drugs have failed simply because chemically based drugs cannot bind to this protein. It doesn't have any binding grooves. So this also means that there's a lot of knowledge around this target, which de-risks our approach because we can benefit from all these papers and all the science around it. Secondly, lipid nanoparticles are well known to be targeted towards the liver. So we know that our LNPs are exclusively going to the liver. We've tested and proven that as well, leaving any other organ untouched entirely with our drug. Of course, we've also shown the broad expression of our intrabody across the liver, and it's safely and stably expressed for over 3 days. And beta-catenin driving resistance to drugs is because what it drives is it prevents T cells to getting into the tumor. And what has been shown by our advisor, Paul Monger, and others is that when you reduce beta-catenin, you can actually get T cells back into the tumor. You can re-engage the adaptive immune system, which is crucially important for cancer progression or inhibiting cancer progression. So we know that with our drug, we can get the immune system back in shape and we can have the body try to heal itself. Of course, we have significantly reduced off-target effects due to the fact that we are using targeted drug delivery and antibodies, and we've already shown repeated and safe administration as well. The ABC program, of course, is our favorite child at the moment, but intrabodies don't stop there. There is ample opportunity going after all these different proteins inside the cell. We won't be able to do all of them, but the opportunities over the next couple of years are massive. There are so many undruggable cancer drivers such as MYC, RAS, or P53 that we can absolutely potentially reach as well. We also can look at the protein level. Working with intrabodies, we can bind on various different modifications on a protein level, be it different splice variants, conformational states such as an on or off state. We can even attack 2 different epitopes on the same protein because of the high specificity and because we work on the protein level, whereas a lot of other drugs that are intracellularly targeted only work on the mRNA level, which limits their targetability. Another interesting field that is viable to this approach is chronic viral infections, such as different hepatitis or long COVID, as we can also bind to the capsid proteins of the viruses rather than binding to a human protein, which makes this approach even safer for applications in chronic infection. Initially, we do need to focus, and we are focusing on liver and lung diseases. The intrabodies are a very complex idea, and the idea has actually been around for a while, but we have spoken to a lot who have envisioned their potential, but it's not an accident that we as a team are the first to make these a reality. As complex as intrabodies are, they also require a complex and multidisciplinary team. It takes a team with scientific, biological know-how to pull all the different parts together. And we have the exact combination that we need between biology, drug delivery, engineering, and strategy necessary to face the challenges needed to develop this modality. We, of course, enormously benefit from our diverse international advisory group that brings decades of medical, industrial, and scientific expertise to the table as well. When we look at exit strategy, we've got some examples here for you that we see as big leaders for us. Investing in well-led companies that are at the forefront of new modalities is highly rewarding. Our goal is to achieve an exit, likely via an IPO, within 9 years of founding the company at a valuation exceeding $500 million US. When you look at the companies here, you can see that these similarly are pioneering companies that have reached very high value exits on similar timelines. But what you can also see is that they actually only had 1 or 2 lead programs in their early clinical trials at IPO. Mermaid Bio has a technology in the team to join the list of Arvinas and BioNTech, and we hope that in 7-8 years, we will be presented just like I present these today. So we really think that we have all the capabilities, and over the last 2.5 years, we truly have built the know-how around intrabodies to make them a medicine and to bring them to the clinic and to the patients. So now it is time to really push this technology and pioneer it. One patient whose lives we really can't change is Adrienne Wilson. Adrienne was diagnosed with HCC at the age of 15, but she died before she was 16. Her sister and guardian, Andrea Wilson, lived through the excruciating misery many patients go through due to the poor treatment options that bring decimating toxicities and limited benefits. Andrea Wilson has created a cancer foundation in Adrienne's honor to advance the search for a cure and to help patients understand their options. She is now one of our advisors because it's key and very important to us to really get the patient perspective and learn early on to really focus our drug development for the patients. All the data, science, and business strategy are just a means to an end, and it's patients like Adrienne that bring us all together today. And it's because of stories like that that we want to make a difference, and we will need your help to do so. And with that, I would like to try and address questions.
Thank you very much, Ranja and Chris. Before going to the intervention of Klaus, let me remind to the people that are here that can ask questions with the Q&A tool or raising the hand. I have some, so we can have a discussion then. And I think that it will appear more questions from the audience. But just right now, Klaus, thank you to be here and have time to explain why you are investing in Mermaid Bio. You have like 5-10 minutes to explain you who is Klaus and why he's investing in Mermaid Bio.
Thanks a lot, Sergi. So first of all, a few words about myself. So I'm a physician by training. After medical school, I spent 4 years as a postdoc at the National Institutes of Health in the US, doing postdoctoral research in atherosclerosis and molecular biology. Then I spent 6 years at Heidelberg University Hospital to receive my training in internal medicine and endocrinology. I still teach in Heidelberg, so every semester I spend a few days teaching internal medicine to students. And then in 2003, I moved to pharma, spent 18 years in big pharma, most of that almost 15 years with Boehringer Ingelheim, including 5.5 years as head of development and chief medical officer. I had the privilege to oversee the late-stage development and approval of a number of drugs. 5 of them are blockbusters now, and they have combined annual sales of more than 20 billion euros now. So I was happy to be part of the team to achieve that. I also have 2 years of commercial experience as a general manager for the UK and Ireland for Boehringer, and then in 2017 moved to Switzerland, joined the executive committee of Ferring Pharmaceuticals as chief medical officer. And now for the last 3.5 years, I'm in this smaller but even more exciting world of startup and venture capital. I'm a partner at a US Switzerland-based venture capital fund. I'm CEO of a company called Ranja, which is a soon clinical stage biotech company, and I sit on currently 4 boards of pharma companies. So yeah, to come back to your questions, what were the reasons for me to invest? And the first one may sound a little funny, but it's rare that you come across an idea where you think, that's such a great idea. Why didn't I have that myself? Or why didn't others have that? So the idea to combine the mRNA technology with the nanobodies to address non-druggable targets that are intracellular, I just found at the time, and still do certainly, very ingenious and very logical. And as I said, I thought, why didn't I think of that? Then you can have 2 reactions. You said, well, if nobody else thought of it or others thought of it and said, this is not good, then you can say, okay, hands off. But, we know some of the biggest companies these days, are based on that first idea, like, Airbnb, Uber, Apple, Amazon, you have it. So I was intrigued, and also it comes on the heels of the mRNA technology being validated through the COVID vaccines. And apologies for that. So on the heels of the mRNA technology validated through the COVID vaccines, and then also I've been very familiar with the nanobody technology because it's been around for quite a while. So I thought it is a great idea, and that really intrigued me. The second thing that I learned in my now 3.5, 4 years of investing into a number of companies is the importance of the team. And I was immediately intrigued and excited about Ranja and Chris. Yes, they're doing this for the first time, so they will have to learn new things, as I had to do as a first-time CEO of my company. But I always found them to be very open to advice, to listen, to be able to adjust based on advice, based on new data. I was very impressed how quickly they build up a network of consultants, advisors, potential investors, scientists that could help them. So that was the second that I learned quickly and helped me to decide to invest. And maybe I'll say one last and third point coming from Big Pharma. I appreciate the importance of intellectual property and the strength of patents, because that's really where the value lies at the end for Big Pharma. And, you know, Ranja mentioned that, you know, at some point looking for an exit, either via an IPO or an acquisition, and then the importance of intellectual property cannot be underestimated. So I was very excited when we early on talked about the IP strategy, and that's obviously several-fold. One is on the technology, combining the mRNA with the LNPs and with the nanobodies. And again, we know from COVID, and BioNTech was on the slide. So BioNTech Moderna, obviously, with that technology, managed to create some IP space and make a lot of money with that. And that's coupled in the case of Mermaid with composition of matter patents, because you can obviously discover and develop nanobodies that have a specific sequence, amino acid sequence, and you can patent that. And this is, as you know, the composition of matter patents are the strongest patent that you can have to fend off competition and make sure that you know, you can have the commercial success after you've appropriately demonstrated the positive benefit risk profile preclinically and clinically. So it's really those three things: the great idea that I still find amazing and ingenious, the strengths of the team, and they're willing to learn and do new things, and the strengths of the IP strategy were the things that led me to invest. I haven't done a lot of investments. Partly because I don't have that much money, but Mermaid was an easy decision for me to take for these three reasons, and I hope that helps.
Klaus, really interesting what you say, and I have a question about one thing that you mentioned. So, again, we remind people that they can ask questions, but we can, Ranja, Chris, if you want, I think we can ask. I can ask you some questions that maybe investors really want to know. One is about competitors. First, direct and indirect. You talk about undruggable proteins. We had several companies in the past treating these undruggable proteins. The first question: why do you think these proteins are better treated with antibodies than small molecules?
I'll start with why.
Yeah.
Why I'll take why the antibodies can target them better, and that small molecules really are limited in what they can bind to. Those chemicals need grooves and pockets to really grab into. If they don't have those, they won't bind really strongly, and when they don't bind strongly, they'll have a lot of off-target effects and bind to other things. Antibodies and nanobodies especially, their binding domains are kind of like fingers. They can really grab into things really tightly, and they're actually really, really long. So they can grab onto a surface, and they don't really need much more pockets to get into. And that's their big advantage. That's been the big advantage with antibodies on the outside of the cell, and that still holds true on the inside of the cell. And we are using nanobodies and single domain antibodies because they are simpler in structure, and they're incredibly stable on the inside of the cell, more stable than an antibody. And I think I'll leave the competitors to you. if you want.
Sure.
Chris, only to make sure, there are someone other people doing intrabodies, and we want to see if there's any company, big, a startup. Why is the standard of care for that?
Yes. So what you have are a number of companies trying to take new technologies to fill in the gaps between small molecules and traditional antibodies. And they're trying a lot of different things. What we haven't seen is a company developing intrabodies the way that we are. In that respect, we've seen a lot of researchers speculate that this might be a useful way to approach things, but somebody really figuring out the problems, they haven't done that. We are the first to really pioneer this modality. What you see in the ways that we think about competition are you comparing our modality to other modalities. So examples of other modalities might be siRNA, for instance, that try to knock down a local protein to a smaller level. There's a comparison there in some of our applications, but there's significant differences. They rely on specific parts of machinery and cells. They rely on the ability of the protein in the cell to degrade over time, whereas ours is very quick to onset and quick to attack a protein. Some of the others would be things like cell-penetrating peptides, or there's another one that's pretty useful, the targeted protein degrader. We haven't really talked about that. But we also have the most recent patent that we have is around turning our intrabodies into protein degraders so that we can target virtually any protein in a cell for destruction, which is something that the kind of traditional TPDs or PROTACs, if you will, haven't been able to really translate. Yeah, I think what I was saying is that we have comparisons to other advanced modalities. There are a number of companies that have already IPO'd in some of these modalities, and we think they're going to have success. But there's a lot of different targets in the intracellular space, and there's some very unique things about the way that we can use intrabodies that they just can't with their modalities. And those are the ones we'll go after, and I think the ones that will really drive our success in time.
Another thing that you explained it, but please, if you can make a reminder, why do your first, as a platform, you can cure or treat a lot of disease with your platform, with your intrabodies? Why to choose first HCCC? It's a strategic decision. It's because it's more easy.
We chose HCC not because it's necessarily easy, because it's actually quite difficult. But first of all, there's a massive target. It's a huge unmet medical need. And beta-catenin is a big undruggable. We knew that we could go after that. And the last thing is that LNPs have a known high liver tropism. So they actually naturally migrate to the liver, over 96% of them. So that targeting aspect is already covered, and we have the safest LNPs available and various different ones available. We do now have our own. So with all of those together, that really is what made sense to us. And the other two minor points are that we can also get orphan drug status with beta-catenin and HCC. That's very important when you look at going to the clinic. And because HCC is so aggressive, there are shortened timelines to the clinic as well. And with beta-catenin, we can expand actually our intrabody to other cancers after that as well. So all of these together really made sense.
Only, Ranja, to make sure, and if you want to explain a little bit what it means to get the drug orphan drug designation in terms of short timings. In case if you are an investor, why it's interesting to have this short timing by the orphan designation?
Yeah, well, I guess there's two real important reasons for orphan drug status. One is that it makes getting to patients and getting exposed to patients much easier and much faster. And that adds value to the product that you're creating. And if things go correctly, then you'll be on the market treating those patients much faster than you would be with the traditional pathway. But importantly for us, having success with our very first intrabody and progressing success there also sells the entire concept and the modality. And so we think the value of Mermaid Bio is going to be well beyond beta-catenin, well beyond orphan drugs, and into the whole space of intrabody applications. The faster we can show their capabilities in humans, the faster the rest of the industry will understand their value, and that'll obviously elevate the company.
Okay, thank you. And what another question we have always for this kind of companies, more biotech, not small molecules, is the way to develop the drug, because it's maybe harder than small proteins. And I think that Klaus talk about. It's difficult to develop this nanoparticle to inject in a body. Do you have any idea of how to do it? It's the same that the one that it's used in the vaccines, as Klaus mentioned. Are you going to contract another company? Are you in contacts? How is the manufacturing working?
Yeah, so we will contract, we will get CDMOs to help us with manufacture the mRNA, but it is significantly easier than small molecules or proteins. With proteins, you have these massive discovery timelines, but then they're easier and cheaper to manufacture, of course. With proteins, you have the problem of having them ultra pure. They have to be pretty much perfect before you inject them. We are really using the patient as the machinery, just like it was during COVID, which really helps significantly actually lower the cost once the mRNA, once you have identified a good candidate. And then to manufacture them, we are doing that on the bench ourselves for our studies. The hardest part is really getting the best candidate, but the manufacturing of the mRNA, we can see that by how many companies are growing like mushrooms out of the ground, working on mRNA therapeutics is absolutely relatively easy and relatively doable to previous technologies. And what's even more advantageous about those is how amenable they are to changes. If you want to have a slight little change, you take the sequence, you change the sequence, you put it back in, but it's such a minor adjustment that can have a major effect on a drug, which previously you had to go and discover a new drug entirely. And that really helps keep costs low and really help in the manufacturing timeline.
Okay, thank you, Ranja. And then, last but not least, can you explain us about the round? Why are you doing this round? Because I think that you have a next round already in negotiations with venture capital. Can you explain the structure of both rounds and new updates for the investors?
Sure, thanks, Serge. We are talking with VCs that we've and starting to assemble what will be our seed round, which will be in the low millions of euros. The goal of that round is to take the beta-catenin program much closer to the clinic, so that when we do a Series A, we can use that funding to completely do a phase 1 study for the anti-beta-catenin program. So we have to step back. So to reach the Series A, we need the seed round. And to reach the seed round is what we're really looking for support from your investor base. This is a really interesting technology, and it's a really exciting technology. And it requires us to tick a lot of boxes before even the VCs want to see what we've done. We've ticked so many of those boxes in the last 2 years, and we've done that with the help of grants from governments, with awards that we've received, with support of WeHi, an institute in Australia who's invested in the company, and then, of course, with support of some of our early supporters, such as Klaus. What we've done in the meantime is demonstrate to the biotech VCs that we need to really impress and take investments from, that intrabodies are a viable concept. And so this funding that we're working on right now with you and your company Capital Cell is to make sure that we have the 6 to 9 months we need to make sure that everybody gets their checks in place so that we can really drive the seed round where it needs to be.
Okay, thank you.
Yeah,
Chris
maybe
and Ranja.
Sergi, if I can add to that,
Yeah,
I talked about the great idea, and unfortunately, all great ideas, sometimes somebody else may come up with it. So this is really about not losing time and keeping the advantage compared to others who may have or have the same idea. So that's why I think this is important to achieve this and get your investor base excited and say, you know, this is such a great idea. We're talking about significant unmet medical need. You saw the example of the 15-year-old girl who unfortunately died. So you don't want to lose time, and for that, it would be invaluable to have the support from investors to move this forward, and not to have to be, you know, not to be forced to stop things for 6-9 months while, you know, all the T's are crossed and the I's are dotted for you know, future investors.
Thank you, Klaus. Really interesting. I think that there's no more questions. I'm going to explain you, or if you want to do it, Ranja, Chris, how these round conditions are fixed like a convertible loan. I don't know, Chris, if you want to start, what are the offers, the convertible loan that you are giving to investors, or if you want, I can explain you by myself, whatever you want.
Oh, sure. I'm happy to. As you can see on our campaign platform page, we're in the process of raising 400,000 euros. We're raising those at an evaluation of 12 million euros. But there's two important caveats to that number. One is that even though these are going to be a share issued through the SPV, they will have a feature that allows them to be re-rated and topped up back to the pre-money valuation of the seed round. Secondly, we are offering a discount on that number so that when you make that first investment, you not only are you have the protection like you would in a traditional convertible note, even though it is a share issue. So this is something that obviously we've already had a lot of support from our existing investors in. We've been on the platform for, I think, less than a week, and we've already seen that number go from sort of 30% to 40% support, which we're very grateful for. So we're excited about just getting that number up to 100% and as quickly as we can, getting back to business and taking this next kind of 3 to 6 months to nail down the seed round.
Thank you, Chris. Let me add something that is in Capital Cell. The last Capital Cell news is that we have a company that is going to be a success, an exit, who is going to give an ROI of 6x to investors who invest in 2019. And we use the same tool as the one that you are proposing, a convertible loan. So it's something that is used in the ecosystem, in biotech investment and all kind of investors, and give really good results, because these investors in 2019 have disclosed this convertible loan and have the chance to invest in the good valuation of the next round as you propose. So it's a tool really interesting, and I have to say that it's been proven by Capital Cell and all the community of investment on biotech.
That's great. Yeah, we think the tool, the approach that you've taken with your investor group, the support that you're giving to biotechs like us is creating success on both sides, I think. And I think that your early work has shown that. And this is the next step in that. We're obviously expecting something like a 20x return. We know that it's a high risk opportunity in some ways, but this is an extremely high value opportunity because of the fact that we are pioneering a whole new therapeutic area.
And last but not least, I share with all the investors the link of the campaign, which they can reserve right now the investment and make sure that they will have this 20% of discount on the next valuation on the seed round. And for me, it's all. I'm gonna say that you have the last minute, Chris and Ranja or Klaus, whatever who won, and we will end the webinar. So only to say thank you very much, Chris and Ranja, to here. Also, really thank you to Klaus to be here. And Ranja, Chris, or whoever who wants has the final minutes.
Well, I think we just want to thank everyone for thinking this through, even if you're considering participating. We're very grateful for your time as well as support, and we're always open to questions as well. And we really stand behind what we do with very candidly, very driven, and very transparently as well. And I hope that comes through in the campaign as well.
So thank you very much, and have a nice evening.
Thank you, everyone.
Thank you.
Bye.
Bye.
Bye.